नाही अजून.
नाही अजून का?
मॅम स्क्रीन हा झाली आता.
हा झाली, रेकॉर्डिंग स्टार्ट झालं.
हा झाली ना.
हो.
2 मिनिटं. आता दिसते सर्वांना?
हो मॅम.
दिसते
Yes,
ना?
ma'am.
टायटल
हो.
काय आहे सांगा?
Evolution of heart in vertebrates.
हा करेक्ट. युनिट नंबर 5 and पेपर नंबर 9. Name of the paper, Comparative Anatomy of Vertebrate. Title, Evolution of Heart in Vertebrates. Then introduction. Comparison, we compare last lecture from the fish to mammal. In fish, how the heart shape, then how this structure, how many chambers, like this way. So the heart is divided into single chamber, 2 chambered, 3 chambered, and 4 chambered. So single chamber heart, it is found in the amphioxus, and it is not a true heart, but below pharynx, the ventral aorta is muscular and it is contractile. It is called as a heart. Then from the fish to mammal, there are some modifications according to their nature and behavior of the animal. So first, in the embryoic condition, there is a cardiac tube is present. This cardiac tube then forms chambers due to the partition or constriction. And then each chamber then divides into 2 separate chambers, and heart gradually shift from head to the neck region or in the thoracic region. So in the fish, heart is present near to the head region, but in the mammal, then reptiles, these are transferred towards the thoracic chamber. Then 2 chambered heart, for example, primitive chordates or protochordate. In primitive chordates, for example, Petromyzon, Myxine, there are in the single line, the heart is present, and these are in 4 chambers. First one is the sinus venosus, second auricle or atrium, third one ventricle, and fourth one small conus arteriosus. Then in dogfish, this is the main. In the fish, we compare fish compared to the reptile, ales, mammal, like this way. So this is the example. Elasmo branch, dogfish is the example. So heart is S-shaped. We see in the diagram, this is the heart, S-shaped structure. In this there is a sinus venosus. How the sinus venosus form? Cardinal vein and hepatic vein, these are together form sinus venosus. Sinus venosus open into the auricle by sino-auricular aperture. Auricle open into the ventricle by auriculo-ventricular aperture. This is the muscular ventricle. Then ventricle opens into the conus arteriosus, and it leads to the ventral aorta. This ventral aorta transfer this aortic arches. And in the dogfish or squalidon, heart is venous heart because it collects impure blood or deoxygenated blood and transfer through this arteries. It is called as afferent arteries. These transfer towards the gill for purification. So these are the chambers, dorsal and ventral views. So heart of squalidon, it is called as a venous heart, and circulation is single circulation. Blood is passed towards the sinus venosus, auricle, then ventricle, then conus artery. It cannot prevent or backward flow. It's showing only single flow. So it is called as a single circulation. So this is the description. Heart is S-shaped. So, multiple question asked on this. Elasmo branch or dogfish, shape of heart, S-shaped, V-shaped, or another leg. So heart is S-shaped. Then there are only 2 chambers. In the dogfish, there are only 2 chambers. These are the auricle and ventricle. These are 2 chambers. And sinus venosus and conus arteriosus, these are the accessory chambers. So blood from heart, it is carried towards the gills for oxygenation. And this type of circulation, it is called single circulation, and heart is called venous heart. Then in the bony fish, in bony fish, for example, labio, conus arteriosus, it is much reduced. Another parts, these are similar. Conus becomes enlarged in that part. There is a consisting conus, there is a muscular wall, and these are inflates like a balloon when the ventricle contact. Then we see in this diagram. So this is the first diagram. Fish. Then in the bony fish. नंतर करायचं लेक्चर. जर 2 मिनिटं थांबा रे.
मेडिसिनची लिस्ट पाहिजे. मेडिसिन काय हो?
हा पीपी किट वगैरे सगळं दिले की त्या
कसं पण ती असल्याशिवाय ही किती आणायची पण
आपण देशमध्ये सरांना विचारायचं.
त्यांना विचारलं ते मग त्या मॅडम कडे गेल्या.
कुठल्या विचार मॅडम?
ते डॉक्टर मॅडम
यादव मॅडम.
हो
होय.
किती आणायची ती
एक तर तात्पुरता आणूया आपण.
काय हँड ग्लोव्स किती आणायचे? आता पीपी किट एक आहे समजा.
हा
पण हँड
ग्लोव्स
ग्लोव्स
एक 10 आणूया.
100 आणायला लागतील.
होय.
हो मास्क किती आणायचे?
मास्क काय कट्टा आणायचा यूज अँड थ्रोचा.
हा ती सॅनिटायझर हे असते.
हा
जे आपलं सर्जिकल मास्क असते.
हॅलो
सॅनिटायझर.
हॅलो हॅलो
येस मॅम. हॅलो.
आहे ना तुम्ही? हा मला वाटलं नेटचा काय प्रॉब्लेम आला.
नाही आहोत आम्ही.
हा हा
किती वाजता येणार?
2 मिनिटं जरा. रूम फ्रेशनर 1-2 आणा. हँड वॉश 1-2. सॅनिटायझर किती पाहिजे? आपल्याकडे आहेच किती सॅनिटायझर? हँड ग्लोव्स 10 आणा. आता तात्पुरते एवढे आले.
नाही परत आणखी मला जायला नको मग ते तुमच्याकडे
तुम्ही आणा. काय तुमच्या मग इच्छेने आणायचं ते आणा. 10,000 काय बसेल ते आणा. हॅलो.
येस मॅम.
हा तर परत सुरू करूया. जरा सर आले होते, काम होतं म्हणून. आपल्या कॉलेजमध्ये हेल्प डेस्क कोरोनासाठी तयार होते, त्याच्यासाठी तयारी झाली आमची. म्हणजे जे संस्थेमधले वगैरे कुणाला कोरोना झाला तर त्याच्यासाठी आपण आयसोलेशन रूम तयार करणार आहोत कॉलेजमध्ये. समजा मुलांना कुणाला वगैरे काय असेल तर त्यासाठी तयारी झाली. ओके. नाऊ सो दिस इज द हार्ट ऑफ dogfish or scalidon. So formation of sinus venosus, these are the ductus qvarian and hepatic sinus. This together form sinus venosus, directly open into the auricle through sino-auricular aperture. This is the auricle, thin structure, open into the ventricle by valve, auricular ventricular valve. Then this ventricle open into the conus. It consisting also semilunar valve, and this joins to the upper side ventral aorta. So this is the found in the dogfish. Then in the bony fish, it is also similar. Only there is a the conus arteriosus. It is muscular. It is called as a bulbous arteriosus. All structure are similar, and these are collected venous blood. So it is in this heart single chamber sinus venosus, auricle, ventricle, and conus arteriosus to ventral aorta. So this blood pass towards the sinus venosus, auricle, ventricle, and conus arteriosus. So single circulation blood, and this heart collected venous blood, it is called as a venous heart. Then in the next diagram, three-chambered heart. Three-chambered heart in dipnoi, amphibia. Dipnoi is the lungfish, amphibia, and reptile. These are the three-chambered heart, 2 auricle, 1 ventricle. So this impure or incomplete separation of the blood. In the amphibian, for example, in the frog. In frog heart, we see in the diagram, this is the frog heart. So auricle, 2 auricle, in between them there is a partition, it is called interauriculum septum. Then in this side, right auricle, there is an opening of sinus venosus. Then ventricle muscular. These two auricle open into the ventricle. It is called as a auriculoventricular valve, left side and right side. So this truncus arteriosus, it opens through this right side, and this consisting spiral valve. And this spiral valve divides conus arteriosus, cavum pulmocutaneum, and cavum aorticum. And this conus upper side, it is called phalangium and synangium. So the spiral valve direct blood towards the pulmocutaneum is lungs and skin, and cavum aorticum is systemic aorta. So this is the muscular ventricle, and this showing this fissure and trabeculae. And in between them, in auricle, there is a partition. It is called interauricular septum, and in this no partition. In ventricle, there is a no partition. So left side blood is pure, which is collected by pulmonary vein, and right side blood is collected, it is impure blood through the sinus venosus. This mixed blood in the ventricle and mixed blood transferred towards the truncus arteriosus to all body parts. So this, it is found in the frog heart. Frog heart is conical in shape, three-chambered and incomplete circulation. And these auriculoventricular valve, these are right side, left side, these are also similar. In the next, this is the dipnoid. So dipnoid, it is the lungfish. In lungfish, sinus venosus in fish and bony fish, these are open posteriorly. But in dipnoid fish, it opens into the right auricle. And left auricle, there is a new circulation is formed. It is called as a pulmonary circulation. And this sinus venosus, there are formation, pre-caval vein and post-caval vein together form sinus venosus. These are open right auricle by sino-auricular aperture. Similar time, this pulmonary veins, this collect blood from the lungs, open into the left auricle. In between them, there is a partition, incomplete partition, interauricular septum and interventricular septum. So there is a mixed blood supply to body parts. So these are the valves are also present in the conus arteriosus. Similar valves are present. So in dipnoid, there is a lung is formed. So pulmonary circulation is, or pulmonary veins are formed. Then in the amphibia, for example, in salamander, the amphibia, there is also right auricle, left auricle, like pulmonary circulation is also formed. But in this, there is a formation of ventricle is single. So mixed blood is transferred towards the body. So, and this conus arteriosus, it is called as a bulbous arteriosus. It is found in the amphibia. So this is the in the reptile. So reptile or lizard. So in the shape of this heart, it is also conical in shape. Right auricle, left auricle. Then there is a ventricle, three-chambered heart, incomplete circulation. So right auricle, there is opening of sino-auricular aperture. All impure blood collected by sino-auricular aperture and open into the right auricle. Then pulmonary vein, which collect blood from the lung, and it is carried towards the left auricle. So in between them, there is a partition, and this is called interauricular septum. Then there is a systemic aorta, and this new pulmonary aorta. This pulmonary aorta carry blood, impure blood towards the lung for purification. This pulmonary aorta opens left side of the right side of the ventricle. But in the ventricle, there are muscular ridges, and due to this muscular ridges, the left side pure blood towards the heart, right side forward this impure blood, and this mixed blood is supplied to the body parts, to the system, because two systemic aorta, and this mixed blood is supplied to the body parts. So auricle open into the auricular. These are the valves are also similar. Sometimes it is called tricuspid, bicuspid. In bird, mammal, it is called. So auriculoventricular or tricuspid, bicuspid. So this is the lizard heart. But in diagram we showing थोडा वेळ थांबा, डायग्राम बघतो. Reptile, then this is the transitional four-chambered heart. Four-chambered heart in the mammal. Bird. In bird, there is also two auricle, two ventricle. Complete separation of auricle and ventricle. Auricle separated by interauricular septum. Ventricle separated by interventricular septum. Then in the right auricle, these precaval, postcaval, these are also opened separately. And it also consisting valve. This is valve is called Eustachian valve. Postcaval open into directly right auricle. So sinus venosus absent in the bird. And left side in the bird there is a presence of pulmonary veins. There are in bird 4 pulmonary veins. These are open commonly. It is carry blood from the lungs. And then auricle open into, these are the valve, these are called tricuspid valve, one side bicuspid valve. And this valve consisting thread-like part, it is called as a cardi tendon. And these muscles only found in the bird and mammal, these muscles are called papillary muscles. So blood from the right ventricle, right auricle to right ventricle, and left auricle to left ventricle. So these are completely separated. Then this pulmonary aorta carry blood from the right side of the ventricle and it opens into the lungs. This carry blood towards the lungs for purification. Then this is the systemic aorta. In bird, the systemic aorta is right side. Only single systemic aorta is present, and this opens through the right ventricle and the left ventricle, and this is the this side there is a pure blood, and this pure blood supplied towards the all body parts. So like this way, this complete separation, auricle separated, also ventricle separated, and chambers are 4-chamber heart. Then in the rabbit also or mammal, there is a separate opening, precaval, postcaval. This is the valve, the Eustachian valve. In this side, pulmonary vein. Then complete separation of auricle, it is called as a interauricular septum, interventricular septum. There is a valve, tricuspid valve, bicuspid valve. In mammal, there is a in between the auricle. In embryonic condition, there is an opening. This opening is called as a foramen ovae. But in adult condition, the only depression is remain. This hole is completely closed, and it is called as the fossa ovalis. And there are also this opening of this sinus venosus externally and internally. There is only depressions are showing, and these depressions are called crista terminalis and sursus terminalis. So these are found in the only mammals. Then like this way also rabbit, bird, all the structures are similar. Only there is the pulmonary aorta which opens to the right side, collect impure blood and carry towards the lungs. And systemic aorta in mammal, there is a left systemic aorta. In bird right side, and in mammal left side. And this collect pure blood and supply to all body parts. So due to the interauricular septum and interventricular septum, blood is completely separated, impure and pure blood, and supplied to all body parts. So in this is the amphibian. So right auricle, left auricle, pulmonary circulation. This is the sinus venosus open into the right auricle, then opening into the ventricle, and this is the spiral valve. The spiral valve consisting in the conus arteriosus, and these are the aortic trunk or aortic arches. In the reptile, there is a separate right auricle, left auricle, then pulmonary. In between auricle, there is a septum, interauricular septum, but in this also septum incomplete. So blood is mixed blood, which is carried towards the left side and supply one aortic trunk carry impure blood, one carry pure blood, and these two systemic aorta then supply to all body parts. And this is the pulmonary trunk carried towards the lung. Then in crocodile, there is a complete 4-chambered heart. So 4 chamber, in between this auricle ventricle there is a partition is present, but This when these two trunks cross each other, in this there is a hole between them, and this mixed blood is supplied towards the body parts. So it is also called transitional heart. In between double circulation and double circulation, there is in between them, so it is called as a, and this hole is called foramina panniza. So in the, this is the mammal and bird. These are the complete separation, right auricle, right ventricle, left auricle, left, there is a complete separation, and this formation of sinus venosus are absent in bird and mammal. Only the pulmonary circulation is also present, and only one aortic trunk or systemic aorta, and only one pulmonary trunk. So these are the, then we compare the how this presence of the in fish, amphibia, reptile, birds, and mammal. So first one is the structure or structure in that pericardium. सगळेजण ऐकतायत ना? हॅलो,
Yes, ma'am.
ऐकताय ना? So first structure is the pericardium. So in all animals or in the chordates, the pericardium is present. So heart is protected inside the pericardium. In there also pericardial fluid. So in the first there is a fish, there is present pericardium present, amphibia present, bird, mammal, like this way. The shape of the heart in the fish S-shaped, in amphibia triangular or conical, in reptile triangular or conical, birds triangular, but in mammal it is pear-shaped, ear-shaped structure. Then nature of the heart, in fish, only fish heart collected venous blood or impure blood, so it is called venous heart. Then amphibia non-venous, mixed blood. In the also reptiles mixed blood. In these birds non-venous heart or complete four-chambered heart. Then chambers, in fish 2 chamber, one auricle, one ventricle. Then in amphibia 3 chamber, 2 auricle and 1 ventricle. In reptile 3 or 4, means in crocodile 4 chamber, 2 auricle and 1 ventricle. In some or crocodile there is a 4 chambers. Then in bird 2 auricle, 2 ventricle. In mammal same. Then nature of circulation, single circulation. In the amphibia, incomplete or mixed blood is present, so it is called incomplete double circulation. In reptile, incomplete double circulation. In birds, double circulation, separate circulation, so it is called double circulation. Then venous and arterial blood. In the fish, it is mixed. Then in amphibia, mixed. All in 3 are mixed blood, but in birds, and these are completely separated. Impure and pure blood, these are separated. Then sinus venosus present in the fish, present in the amphibia, present in reptile, birds and mammal. Sinus venosus is absent. Sometimes question asked on this. Sinus venosus absent in dash dash animal or chordate, fish, reptile, like this way. So sinus venosus absent in bird and mammal. Then conus arteriosus present in fish. Then conus or truncus arteriosus, it is present in amphibia. In reptile, conus arteriosus absent. In bird also absent, and mammal also absent. Then truncus arteriosus, it is present in fish, in also amphibia. Then absent in reptile, birds and mammals. Then spiral wall absent in fish, present in amphibia, then absent in all reptile, bird and mammals. Then interauricular septum, it is absent in the fish, present in the amphibia, present in reptile, aves and mammal. Then semilunar wall, these are present in all animals. Semilunar walls are present. Bicuspid, tricuspid wall, these are absent in the fish only. In fish there is a presence of semilunar wall, and in all animals the semilunar walls are present. In amphibia, reptile, birds and mammals. Then precaval and postcaval vein. In fish there is a hepatic sinus or ductus queris veins are present and sinus venosus form. But in fish, precaval and postcaval absent. In amphibia, these are open into the sinus venous. Two precaval and one postcaval together form sinus venosus. In also like this way, these are present in reptile but absent in mammal. In mammal, precaval and postcaval, these are separately open. And in also bird, similar. Then foramina paniza, absent in the fish, then absent in the amphibia, only present in the reptile and in crocodile. So birds, these are foramen paniza, and mammal, it is also absent. Foramen paniza means two aortic arches when cross each other. At that time place, there is a smaller hole, and this hole it is called as a foramen paniza. And mixed blood is found only in the crocodile. So it is called as a transitional heart. Then pulmonary vein absent in the fish. Open in the amphibia, there is a pulmonary circulation start. So left auricle opens. Then in the reptiles, open into left auricle. Then in also birds, there are four pulmonary veins are present. These are open into the left auricle. In mammal, two pulmonary vein open into the left auricle. Then blood vessel arising from the ventricles. So these are the conus arteriosus, where the blood vessels originated. Then truncus arteriosus. In the reptile, there are three aortic arches: pulmonary aorta, right systemic and left systemic. In bird and mammal, there is only one systemic, only one pulmonary aorta. And in mammal also, there is a presence of only one pulmonary aorta and left systemic aorta. So like this way, these are the characters or structures found in the compare. We compare like this way, which parts are absent, which parts are developed. So mammal and birds, these are well developed parts. But in the fish, amphibia, fish, these are some parts are developed, some are not well developed. So in amphibia, these are well advanced than the fish, reptile well advanced than the amphibia, birds well advanced than the reptile, and mammals, these are also well advanced than the birds. Like this way we compare like this. So another also characters in that, how this pericardium, where this heart is located. Heart is located in fish mid-ventrally and beneath the pharynx region, and is inside the pericardial cavity. In amphibia, it is beneath the in amphibia, heart is located in thoracic cavity. In reptile, it is also thoracic cavity. In owls, it is thoracic cavity, and in mammal, it is also thoracic cavity. Pericardium, how it is formed? It consisting two-layered membrane, and two-layered membrane, inside them there is a pericardial fluid which protect heart from the mechanical shock. Size and shape we see. Color of the heart: in fish, reddish color or brown color. In amphibia, reddish color. In reptile, reddish color. In owls, also reddish color. And in mammal, also reddish color. Then chambers, how many chambers? In fishes, linear chamber, sinus venosus, auricle, ventricle, conus arteriosus. Only two chambers are auricle and ventricle. Another two chambers are accessory. So then in amphibia, two auricle, one ventricle. These are not demarcated externally. Sinus venosus and truncus arteriosus also present in amphibia. But in reptiles, two auricle, one ventricle, ventricle incompletely divided, and sinus venosus also present. In owls or bird, four chambered, two auricle, two ventricle, ventricle not distinguishable externally. In mammal also similar. Then sinus venosus triangular in shape. In fish, which receives venous blood from the body by formation of two ductus querian and to hepatic sinus, formation of sinus venosus. In amphibia, it is triangular. Sinus venosus is triangular and open into the dorsally to right auricle and receive venous blood from the two precaval from the anterior side and one postcaval from the posterior side. In reptile, sinus venosus is bilobed structure and it opens into the dorsal side of the auricle formed by two precaval and one postcaval. In bird, sinus venosus absent, only three caval veins open directly, postcaval and precaval open into the right auricle. Then sinus venosus in mammal, it is right auricle merged and externally marked by sulcus terminalis, internally marked by crista terminalis, and there is sinus venosus absent. Then sino-auricular aperture, sinus venosus opens into the posterior side of the auricle by opening sino-auricular aperture. It also guarded by wall. These are all in similar in amphibia, in reptile, in aves, and in also mammals. But in the mammals, the postcaval vein which is guarded by eustachian wall. In bird also, right auricle, postcaval vein guarded by eustachian wall. Then auricle is triangular in fish, auricle rectangular in also amphibia, auricles divided completely by interauricular septum in reptile, birds and mammals. Auricle wall is thick, thin spongy in fish, thin without muscular process in amphibia, then thin network of low muscular ridges in the reptile and thick walled in the aves and mammal. So in the aves and mammal, muscular ridges inside the auricle, it is called as a musculi pectinati. So MC question asked on this, in which animal musculi pectinati is present. Then pulmonary vein it is absent in the dogfish or scalidon. Frog, common pulmonary vein open into the left auricle. Then lizard also opens into left auricle. Then in bird, four pulmonary vein open common aperture into left auricle, and in rabbit or mammal, two pulmonary vein open into the common opening left auricle. Then AV aperture or wall. In this also, auricle open into ventricle by AV auriculoventricular aperture guarded by membranous wall. In frog also similar. In lizard also similar. In bird mammal it is called tricuspid and bicuspid wall. Ventricle in fish thick, in also frog thick, lizard also thick, and mammal and bird also it is thick in nature. Columnicorni, these are the muscular ridges, these are called columnicorni. These are absent in the fish, then these are found in the frog, then lizard, and in also pigeon and in also rabbit or mammal. These are found. These are called papillary muscles. Papillary muscles are only present in the mammal and bird, not in other animal. Conus arteriosus, it is present in the fish and amphibia. Then conus are absent in the lizard, absent in pigeon, and absent in also rabbit. Aortic arches, these are in fish, 5 pairs of aortic arches, and in amphibia, the truncus arteriosus divide into right and left. These are common carotids, systemic and pulmocutaneous. In lizard, it divided into pulmonary right and left systemic and cross each other. Then ventral, in bird and mammal, only 2 aortic arches are present. Right opens through the right ventricle pulmonary and left ventricle right systemic. In bird, mammal, pulmonary one and systemic one. These are only present. Then foramina pannisa, it is only present in the reptile. A working of heart, in fish, heart receives venous blood, send it to gill for aeration or purification, and this heart is called venous heart, and circulation, single circulation. In frog, heart receives oxygenated, deoxygenated blood and supplies mix the blood to the different body part. It is also transitional single circulation, incomplete circulation. Then mixing of venous and oxygenated blood incompletely divided by the ventricle, it is also single circulation. In bird and mammal, there is a double circulation because heart completely four chambered without mixing of venous and uh, oxygenated blood. So in the bird and mammal, there is separately circulation, and this circulation it is called as a double circulation because pure blood and impure blood, these are separately uh, separately supplied to the body parts. समजलं का ग? हॅलो.
Yes, ma'am.
हा, आता ह्यातले एमसीक्यू क्वेश्चन विचारतात त्यावरती बघा. सायनस व्हेनोसस कशावर कुठल्या एनिमल मध्ये आहेत? नंतर फॉल कुठल्या कुठल्या आहेत? फोरामिना पॅनिजा कशामध्ये? कशामध्ये फोरामिना पॅनिजा कुठल्या एनिमल मध्ये आहे सांगा?
Reptiles.
रेप्टाइल्स मध्ये फक्त क्रोकोडाईल मध्ये फक्त ज्या ठिकाणी क्रॉस होतात ॲवार्टिक ट्रंक सिस्टेमिक लेफ्ट अँड राईट, त्यावेळेला तिथे एक होल असतो, त्या होलला म्हणतात फोरामिना पॅनिजा, आणि त्यामुळे कम्प्लीट ऑरिकल सेपरेटेड असते, व्हेंट्रिकल सेपरेटेड असते. मात्र ज्या वेळेला क्रॉस होतात तिथं होल असते, त्यामुळे मिक्स्ड ब्लड हे बॉडीला सप्लाय केले जातात. फक्त क्रोकोडाईल रेप्टाईल मध्ये फोरामिना पॅनिजा असतात. आलं लक्षात? नंतर व्हेनस हार्ट, विच हार्ट इज कॉल्ड व्हेनस हार्ट? कुठल्या एनिमलच्या हार्टला म्हणतात व्हेनस हार्ट?
Scolodon.
स्कॉलिडॉन. किती चेंबर्ड आहेत? हाऊ मेनी चेंबर? स्कॉलिडॉन मध्ये किती चेंबर असतात? आता सांगितलं ना मी, ट्रू चेंबर 2. समोर दिसतं की चार्ट मध्ये बघा, चार्ट आहे का समोर? हाऊ मेनी
Yes,
चेंबर?
ma'am. 2 chambers.
2 चेंबर. त्यामध्ये 1 ऑरिकल 1 व्हेंट्रिकल हे ट्रू चेंबर झाले, आणि दुसरे जे आहे सायनस व्हेनसस आणि कोनस आर्टेरिओसस हे असेसरी चेंबर्स आहेत. आता यामध्ये यातले तुम्हाला आता आणखी यातले क्वेश्चन कुठले विचारतात बघा. इंटर ऑरिक्युलम सेप्टम कुठल्या एनिमल मध्ये नाही सांगा आता? इंटर ऑरिक्युलम सेप्टम, ऑरिकलच्या मध्ये जे सेप्टम आहे ते कुठल्या एनिमल मध्ये नाही? बर्ड मध्ये आहे का? मॅमल मध्ये?
Fish आणि amphibian.
फिश मध्ये नाहीये.
फिश मध्ये नाही. हा, इंटर ऑरिक्युलर फक्त फिश मध्ये इंटर ऑरिक्युलर सेप्टम नाही. इंटर व्हेंट्रिक्युलर सेप्टम कुठल्या कुठल्या एनिमल मध्ये नाही?
फिश मध्ये
हा,
एक मिनिट मॅम. हा, फिश मध्ये नाहीये, फ्रॉग मध्ये नाहीये,
हा,
आणि रेप्टाइल्स मध्ये इनकम्प्लीट आहे.
हा, म्हणजे त्यात फिश, रेप्टाईल, अँफिबिया मध्ये हा इनकम्प्लीट आहे, म्हणजे इंटर व्हेंट्रिक्युलर सेप्टम नाही. आहे कुठल्या दोन एनिमल मध्ये? बर्ड आणि मॅमल.
मॅमल्स.
हा, आता बायकस्पीड आणि ट्रायकस्पीड वाल कुठल्या कुठल्या ह्याच्यात आहे? बायकस्पीड आणि ट्रायकस्पीड
रेप्टाइल्स
सगळ्यामध्ये आहे, सगळ्यामध्ये. फक्त फिश मध्ये
ऍबसेंट आहे.
हा, फिश मध्ये कुठले आहे? सेमिल्युनार आहे फक्त, आणि बाकी सगळीकडे मग आहे. आता इथं सायनस व्हेनोसस फिश मध्ये कसा तयार होतोय? डायग्राम मध्ये दिले बघा. सायनस व्हेनस हाऊ इट इज फॉर्म. 2 डक्टस क्युइरा आहे आणि हिपॅटिक सायनस मिळून सायनस व्हेनोसस तयार होते. बाकीच्या एनिमल मध्ये 2 प्री कॅव्हल आणि 1 पोस्ट कॅव्हल. 2 प्री कॅव्हल म्हणजे हेड रिजन कडून ब्लड कलेक्ट करून येणाऱ्या, आणि म्हणजे दोन्ही बाजूच्या 2, आणि पोस्टेरियर साइड कडून येणारी पोस्ट कॅव्हल, हे तिन्ही एकत्र येऊन सायनस व्हेनोसस हा सगळ्यामध्ये तयार होते. कुठल्या एनिमल मध्ये सायनस व्हेनोसस कुठल्या कुठल्या एनिमल मध्ये आहे? अँफिबिया, रेप्टाईल, बर्ड आणि मॅमल मध्ये नाही. त्यामुळे इथं फक्त प्री कॅव्हल आणि पोस्ट कॅव्हल. आता बर्ड आणि मॅमल मध्ये काय होतंय? दीज आर सेपरेट, ओपन सेपरेटली. म्हणजे प्री कॅव्हल आहेत मात्र त्या आपल्या आपल्या वेगळ्या ओपन होतात. पोस्ट कॅव्हल आहे मात्र आपल्या आपल्या वेगळ्या ओपन होतात. आता बर्ड आणि मॅमल मध्ये हार्टला जी सप्लाय करणार आहे त्याला कोरोनरी सायनस म्हणतात, आणि व्हॉल्व जी आहे त्याला थेप्सियन व्हॉल्व म्हणतात. फक्त मॅमल मध्ये हार्ट कडून ब्लड कलेक्ट करून घेणारी आणि त्याला कोरोनरी सायनस, आणि व्हॉल्व जी आहे त्याला थेप्सियन सायनस. ह्याच्यावरती पण क्वेश्चन विचारला जातो. अजून काय शंका आहे का तुम्हाला? आता लिहायचं कसं हे समजलं का? आता मी एक क्वेश्चन तयार करून तुम्हाला दिलेला आहे हार्टचा, ह्याचा ब्रेनचा, होय की नाही?
हो.
हा, तसं आता ह्याच्यावरून हे सुद्धा तुम्ही अब्सेंट प्रेझेंट असं म्हटलंय ना. आता समजा इथं कम्पेअर दिलं फिश आणि रेप्टाइल्स, कसं कम्पेअर करायला सांगा मला. पहिले कॅरेक्टर काय ना, पेरिकार्डियम इज प्रेझेंट इन फिश, इन रेप्टाइल. शेप ऑफ हार्ट एस शेप, इकडे काय येणार ट्रायंग्युलर शेप. नेचर ऑफ द हार्ट इकडे फिश मध्ये व्हेनस हार्ट, इकडे कसं येणार नॉन व्हेनस हार्ट. चेंबर फिश मध्ये 2 चेंबर, रेप्टाइल मध्ये 3 or 4 चेंबर. नेचर ऑफ सर्क्युलेशन सिंगल सर्क्युलेशन, इकडे इनकम्प्लीट डबल सर्क्युलेशन किंवा मिक्स्ड सर्क्युलेशन. व्हेनस आर्टेरियल, व्हेनस अँड आर्टेरियल ब्लड इथं आहे का? आहे. फिश मध्ये मिक्स्ड आहे, इकडं अँफिबिया ह्याच्यामध्ये मिक्स्ड आहे. कोनस आर्टेरियोस आहे का? फिश मध्ये आहे, रेप्टाइल मध्ये नाही. असे हे चार्ट किती 15 पॉइंट्स किती होत आहेत बघा, 18 होत आहेत. ह्या 18 पॉइंट्स झाले की डायग्राम वर आता उलट्या दिलेल्या आहेत ना त्या, ती डायग्राम वर द्यायची आणि ह्या चार्ट नुसार हे सांगायचं. एवढं झाले की तुमचा हा फुल क्वेश्चन तयार होतो. समजलं?
येस मॅम.
हा, मग आता तुम्ही ह्याच्यावरती तुम्ही तुमच्या इच्छेने, जसं पहिले केलं होतं ना हार्टचं ह्याचं ब्रेनचं, तसं तुम्ही तुमच्या इच्छेनं कुठलेही 2 कंपॅरिझन तयार करायचे. आता डायग्राम घेतलेत, फोटो काढ, स्क्रीनशॉट काढलाय का तेजी? स्क्रीनशॉट काढून प्रेझेंटीचं पण सांग मला किती आहेत काय. प्रेझेंट किती आहेत ग?
24 मॅम.
24 का? हा, आणि हा चार्टचा फोटो काढ, स्क्रीनशॉट काढून ठेव.
येस मॅम.
बर, आता मी माझं लेक्चर, आता मला जरा काम आहे ते आपल्या कॉलेजचं म्हटलं ना मी आता, हेल्प डेस्क तयार करायची ती कोरोना
हा
सेंटर
मॅम.
साठी. म्हणजे आपण आयसोलेशनला लोक इकडे आणणार आहेत, ते जरा तयारी आहे, त्यामुळे मी आता इथं थांबते.
येस मॅम.
आता दुसरं घेणार होते मी, पण आता ती लोक यायला लागली, त्यामुळे मला थांबावं लागेल.
मिस मग आता कुणाचं
फक्त
लेक्चर आहे का?
आता बहुतेक नसावं, कारण सर आता पाऊण वाजलायच.
हा.
जर सर आहेत प्रॅक्टिकल एक्झाम मध्ये, कोळी सरांना तर कोरोना झालाय, त्यामुळे कुणीच नाहीये लेक्चर घ्यायला आता. त्यामुळे तुम्ही तुमचा स्टडी करा. हे जे दिलेलं आहे हे मी चार्ट फोटो काढून घ्या, ह्याच्यानुसार तुम्ही तयार
येस
करून
मॅम.
ठेवा तुमच्या नोटबुक मध्ये.
येस मॅम.
ओके, थँक्यू.
थँक्यू मॅम.
हा, चालते, वेलकम.